Assalamu alaikum. I welcome you all in this video. In this video, we are going to discuss about the cell cycle, what it is, and how it's going to proceed. So, as we already know, the cell cycle is mainly composed of G1, S, G2, and M phase, and there is another phase which is followed by M, and this is generally present over here, and it is called as G0 phase. So in G0 phase, cell remains in the quiescent phase, so it exists, but it's not going to enter into the round of cell division, and that phase is called G0 phase, right? So as you can clearly see, this orchestra of G1, S, G2 and M phase is actually started by G0 phase, and the cell prepares itself to go for a complete round of cell division. Now once the cell enters in G1 phase, there is a point which is called a decisive point, or where this is generally called as R point. The reason R is labeled as restriction point. From here the cell makes up its mind that now it is going to complete this whole round of cell cycle. Alright, so the period during which the cells are responsive to mitogenic growth factor signals are to anti-mitogenic growth factor signals is like transforming growth factor beta. This is anti-growth factor, and this is growth factor signal, is elaborated in this green portion. So once the cell enters this restriction R point, irrespective of whether you are introducing serum starvation or you are depriving the cell from any extrinsic growth factor or anti-growth factor, now cell will try its maximum to complete the cell cycle. Now obviously intrinsic factors like hypoxia, stress do influence, but by and large, once the cell crosses this barrier of R point, it is going to complete the round of cell cycle. Mainly, the cell cycle is completed by CDKs. Now, CDKs, as the name indicates, are cyclin-dependent kinases, but to activate these cyclin-dependent kinases, we need cyclins, right? Now, there is a short form of these cyclins. I have labeled them over here for the A's to memorize. Now, if we remember D, E, A, B, now in each phase this is the sequence of cyclins. Now, cyclin D1 forms a complex with cyclin D dependent kinase 4R6, and this facilitates for early and mid G1 phase, followed by the activation of cyclin E forming a complex with CDK2. This facilitates the entry from R point to the S phase. Then there is an overlap of cyclin dependent kinase 2, and this CDK2 is actually been forming a complex with cyclin A, so E is being replaced with A, followed by CDC2. This is also called as cyclin dependent kinase 1, but for the A's of the same flow we are giving it as CDC2, forming a complex with cyclin A, and then till mitosis there is an overlap of cyclin B forming a complex with CDC2. So the name, as the name indicates, that the cyclins, why they are called cyclins? The cyclins are basically the proteins, but their expression is in cyclic orientation. So if we focus on this cyclin B expression, for example, here you can clearly see that in each mitosis phase, the expression of cyclin is higher as compared to the remaining phases of the cell cycle. And if we go for an overall expression pattern of all these cyclins, if we start our expression, the highest expression of cyclin D1 is high over here. Now I have mentioned the word D1. That means there are few isoforms of cyclin D. Now these isoforms are D1, D2 and D3. So D1, D2 and D3. Okay, so this is D1, D2, um, this is D2 and D3. Okay, this is D2, D3, and the highest concentration of D cyclins is high in G1 phase. Then there is a reduced expression of cyclin D, followed by an upsurge later on in again the G1 phase. While cyclin B is already been described as shown in the figure above, and cyclin E expression is short-lived, but it is present between the late G1 phase and is tapering down in the SR synthesis phase of the cell cycle. Now, the question is that there are multiple growth factors. All these factors, including hedgehog pathway, various ligands, cytokines, growth factors, HER2-like things, they all are converging to increase the expression of cyclin D1 by binding at its promoter region. For example, this growth factor binds with SOS followed by RAS-RAF pathway, FOS and AP complex, FOS and Jun merge together to form a heterotrimeric complex of AP1 protein which binds with D1 promoter. Now that's why this factor is binding with D1 to initiate the process of production or transcription of D1 messenger RNA so that D1 protein is synthesized. That is going to bind for cyclin dependent kinase complex 4 and 6 over here. So all the growth factors, mitogenic factors are actually trying to produce cyclin D in sufficient quantity so that they can activate CDK4 and 6 so the cell can pass this G1 mid phase. Now depending upon the variability of these growth factors, there are D1, D2 and D3 cyclins. So if we look at this table, we can clearly see that estrogen receptor and some other important extrinsic mitogenic factors are being entertained by D1 cyclins, while mitogens and interleukins are being integrated by are trying to be regulated D2 and D3 promoters to increase the message. And lastly, this is a comprehensive explanation that the program influenced by the extracellular signals is mainly dealt till this point, that is early E, I'm just labeling E over here, early and mid phase of G1 phase. Okay, once this has been crossed, this R point has been crossed, this cyclin E produced integrates with CDK2. Now, irrespective of whether we starve the cell or we expose them to any more mitogenic factors, cell is not responsive to the extrinsic source. Now they are going to regulate this orchestra by using the internal intrinsic factors. That's why the term cell autonomous programming is used in this phase. So CDK2 interacts with cyclin E. Now this cyclin E is actually been facilitated or being produced by formation of this complex. And then this CDK2 itself integrates with cyclin A, and this complex binds and process for the activation of CDC2 and cyclin A. And then there is followed integration of CDC2 with cyclin B. Again, during mitosis phase, this cyclin B is going to be reach its peak, and then it's going to taper further, and there is a sharp decline of cyclin B. So this whole orchestra is how the regulation of cyclins interacts with cyclin-dependent kinases. So if you remember any of these images, that is pretty straightforward thing. Now the most interesting point to understand is how these cyclins are being communicated to go up and down regulated. The whole orchestra is being tightly regulated by the retinoblastoma protein. That is being elaborated in my next video. Thank you very much for your attention and your interests. Similarly, like every yin and yang, you came to know that cyclin-dependent kinases also have an inhibitors. So the word IN right from here, NK from here. So far, there are 7 CDK inhibitors being identified. Number 1, P16, P15, P18, and P19. They are primarily targeting cyclin D and CDK4 and 6, followed by the remaining 3 are primarily targeting the remaining cyclins and cyclin-dependent kinase complex. Okay, so these 7 inhibitors, and this is the nomenclature. So IN from the inhibitor, K from the kinase. So all these are targeting cyclin-dependent kinase 4. That means this 4 has a more pivotal role as compared to 6. But this complex is actually been targeted by the both CDK6 and D is being influenced by all these 4 members. Similarly, P27, P21 and 57 also have a distinct role to address the remaining rounds of cell cycle. Okay? If you have any questions, please feel free to ask. But the regulation of these inhibitors is also governed by the phosphorylation of retinoblastoma protein. Hope you understand and like the video. Thank you.